So let's check question number 131 until 136. So your first question 131 is, the first step of photosynthesis is, you know that photosynthesis consists of two types of reactions. Okay, the first one is light reaction and then it's a dark reaction. So when you say photosynthesis, it consists of light reaction and dark reaction. Okay, light reaction supplies ATP and NADPH2, which is utilized by the dark reaction, okay, for the process of generation or fixation of the carbon dioxide. Instead, from the dark reaction, okay, the light reaction gets ADP. What it gets? ADP and NADP, right? Which is further converted into ATP and NADPH2. Okay, so the cycle continues. So the first thing is it's a light reaction, okay, and not a dark reaction. Okay, so let's see. And when you talk about light reaction, okay, and light reaction consists of cyclic photophosphorylation, okay, and non-cyclic photophosphorylation. So non-cyclic photophosphorylation gives you the NADPH2 as well as the ATP. Okay, it consists of photosystem. There are two photosystem, photosystem 2 and photosystem 1. Photosystem 2 runs on around 680 nanometer wavelength, okay, and it runs on around 700 nanometer wavelength, okay, and when the sunlight, okay, strikes on it, the photosystem, the electron gets excited, okay, which is captured by the primary electron acceptor. And since the electron is having high amount of energy and the step by step release the energy and comes to a ground state, okay, it reaches the PS1, and PS1 against the light energy strikes, okay, and re-excite the electron. Those electrons are used to reduce NADP into NADPH2. And while this process is happening, the energy of the electron is used to synthesize, okay, ATP, which will be considered as substrate level phosphorylation. Okay. Okay, then what happened? So since the electron is lost by the PS2, okay, and those electrons have to be replenished or restored, and it is done by the splitting the water molecule. So water molecule gets split because of the light. It's called photolysis of water. And as a result of photolysis of water, it generates 2 electrons, 2 protons, and oxygen. Those oxygen release, which is consumed by us, okay, or maybe used in aerobic respiration. And the electron goes into the PS2 system, okay. So PS2 system will accept the electron only and only when it get oxidized, okay. Otherwise, it will not accept the electron, okay. So first, it has to get oxidized, okay. So if we see the sequence of reactions, okay, and let's see the options. So excitation of electron of chlorophyll by a photon of light, it's true, okay. Formation of ATP, ATP can be formed only when the electrons get excited and it moves from higher energy level to lower energy level, and while moving from higher to lower energy level, the energy is used to synthesize ATP. Okay, so it will be the next after the excitation of the electron. Okay, then attachment of carbon dioxide, okay, to 5-carbon sugar. They are mentioning the dark reaction, and dark reaction only can happen after the light reaction is completed. Then ionization of water, which is also known as photolysis of water. Photolysis of water, okay, is useful only and only when the PS2 system lost its electron, okay, and it will only lose, okay, when they are excited by the sunlight, okay, or we can call it as a photooxidation, okay, because resulting in the discharge of electron, okay. So your right option will be option A. Then question number 132. The chemiosmotic coupling hypothesis of oxidative phosphorylation proposed that adenosine triphosphate, that's ATP, is formed because high energy bond are formed in mitochondrial protein. It's false. Okay. ADP is pumped out of matrix into the intermembrane space. Okay. It is not pumped out. Okay. A the proton gradient forms across the inner membrane. Okay, across the inner membrane, it's true. Okay, there is a change in the permeability of inner mitochondrial membrane towards the adenosine diphosphate. Okay, so this is also incorrect with respect to the statement given. Okay, because the statement said chemiosmotic coupling. Okay, so the coupling process happens because of the generation of a proton gradient. Okay, now how the proton gradient is generated? Okay, so if you remember your Krebs cycle, okay, during the Krebs cycle, or we can also call it a TCA cycle, the system is generating, okay, NADH. Yeah, they're generating NADH. Okay, and this NADH and also generating FADH. Yeah, NADH2 I will write, and FADH2. Okay, now in the mitochondrial membrane, inner membrane, there are complexes. Okay, there is complex 1, complex 3, then we have the complex 2, okay, and then there is complex 4, and then we have the oxysome. Okay, which is complex 5 you can consider, or can consider as an oxysome, which is F0 and F1 particle. Okay, so what happened? This NAD, okay, loses its electron, okay, or get oxidized, right? Get oxidized into NAD. Okay, and then they come to the Krebs cycle and again get reduced into NADH2. Same thing happened for the FADH2. FADH2 goes to the complex 2, okay, where it get reduced, sorry, it get oxidized into FAD, okay, and then in the TCA cycle, it again get reduced into FADH2. The electron, okay, which is captured by complex 1 is given to complex 3, and while it does so, it pumps the proton. Okay, complex 1, complex 3, and complex 4 are proton pump. So they are pumping the proton from the matrix, okay, into the inner membrane space. Okay, we have the inner membrane, and here we have the outer membrane, and the space between the inner membrane and outer membrane is the intermembrane space. So as more and more protons are pumping, while the electrons are shifted, okay, from one complex to another complex, okay, it generates a proton gradient. So since there's a huge amount of proton gradient generated, okay, this gradient used, okay, to generate ATP while they are passing through the oxysome. When they pass through the oxysome, okay, oxysome use that energy to convert ADP into ATP. Okay, so your right option will be option C. Then let's see the next question. Question number 133. Okay. So it says that a tall true breeding garden pea plant is crossed with dwarf true breeding garden pea plant. Generally, for a recessive trait, okay, recessive trait is only visible when they are in homozygous condition. Okay. So since they are using the word true, so I consider that. Okay, let's consider that the plant are in a homozygous condition. Right. So if you cross them, you already know by the Punnett square. Okay, you are going to get what you're going to get. Okay, you are going to get like this. Okay, and you will be have a genotype of this. Okay, so it will be 1:2:1 ratio, which means you will have 1, then 2 heterozygous condition, and then 1 homozygous condition. Okay, so we have 1 homozygous dominant or homozygous tall, 2 heterozygous tall, and 1 dwarf. Okay, so the ratio or the option suggesting here is option A. Okay. Then your next question. Okay. The next question is, which of the following is not applicable to RNA? Not applicable to RNA. Okay. So first thing is it says that 5' phosphoryl and 3' hydroxyl group N. Do they have? Yes, they have. Okay. If you remember your structure. Okay. Okay. So this is first carbon, second carbon, third carbon, fourth carbon, and there is a fifth carbon, right? So second carbon contain hydroxyl group, third carbon also contain hydroxyl group. So this is 3 prime, and fifth, there is a phosphate here, right? So it will be 5 prime. Okay. Then heterocyclic nitrogen bases. Yes. Okay. They have purines and pyrimidines and pyrimidines, which is AG. Okay. And then they have the UNC. Okay. Since they are the RNA, so they have. Okay. Do they follow Chargaff rule? In general, they don't follow, because Chargaff rule says that purines. Okay. The quantity of purine is equal to the quantity of pyrimidine. But this is applicable if they are double standard. Okay, like you have a A here, then you have a U here, you have U here, you have A here, you have G here, you have C here, you have C here, you have G here. So if you calculate the quantity of purine in pyrimidine, it always stays same. But this is applicable for DNA, it's not applicable for RNA. Okay, RNA is negatively charged. Yeah, true. Our RNA is negatively charged. They have phosphate group, so they are negatively charged. It's true. Okay, so it's not applicable statement is Chargaff rule. Okay, Chargaff rule is not applicable for RNA. If in case, if you in future, if you see some question which says that is applicable, then is applicable only and only if the RNA is a double standard RNA. Okay, if the RNA is double standard, then you can say that the Chargaff rule is applicable. For single strand RNA, when you talk about RNA as general, okay, RNA is a single stranded. So for single strand RNA, Chargaff rule is not applicable. Okay, and then here's a question that transfer of yeast cells, okay, from aerobic condition, from anaerobic condition to aerobic condition. Okay, so yeast are in anaerobic condition. You are shifting the yeast from anaerobic to aerobic condition. So they are in anaerobic. You are shifting them into aerobic. Okay, so what will happen? Yeah, they are started consuming CO2. Yeah, auto is consuming. So how it's going to change? What changes that can be observed in yeast cells? It's interesting question. Okay, so see, for example, okay, for per glucose molecule, yeah, one glucose molecule in anaerobic condition, they are going to generate what they're going to generate. They're going to generate 2 ATP molecule, net gain 2 ATP, right? They generate, of course, they generate 4 ATP, but 2 ATP is used, okay, as a investment phase. Okay, investment phase, they're using 2 ATP when they convert glucose into glucose 6 phosphate and fructose 6 phosphate into fructose 1,6 bisphosphate. Okay, in two places, ATP is consumed. Okay, then ATP is produced. Okay, per triose for glyceraldehyde. Okay, 2 ATP produced. Dihydroxyacetone phosphate will convert it into glyceraldehyde phosphate, which again going to convert to pyruvic acid, will again generate 2 ATP. So 2 plus 1 plus 1, 2 ATP will be net gain. Okay, so they get 2 ATP as a net gain. But if you consider aerobic respiration, okay, there is 2 ATP from glycolysis and then 36 ATP from the mitochondria. So in total, 38 ATP per glucose molecule per glucose can be generated. Since they are generating so much amount of glucose, so utilization of the sugar, okay, will be less. Let me tell you how. Okay, if they need 36 ATP, for example, the yeast needs 36 ATP for running a process. Okay, if the process is from anaerobic, they need to consume how much? They need to consume 38 ATP. Let's consume 38 ATP. Okay, for our easy maths, 38 ATP. So they'll need to consume 19 glucose. Okay, since one glucose generates 2 ATP, so 19 glucose molecule to generate 38 ATP. But if their aerobic respiration running on, so one glucose molecule will give them 38 ATP. Okay, so here they are using less amount of sugar to generate the same amount of ATP. Here they are using more amount of sugar. Okay, condition one. Condition two, decrease in CO2 evolution. Okay, CO2 when they are doing complete oxidation, okay, inside the mitochondria, the glucose will be completely oxidized and completely decarboxylated, generating 6 molecules of CO2. Okay, so CO2 will be generated. So comparing to anaerobic condition, in aerobic condition, the quantity of CO2 will be increased. Okay. Then decrease in ADP per unit time. Yes, of course, because ADP is getting converted into ATP. Okay, so if there was 6 ADP, it will gradually become 5 ADP, 4 ADP, 3 ADP, 2 ADP, and 1 ADP, and 0 ADP. And whereas 1 ATP will become 2 ATP, 2 ATP will become 3 ATP, 3 will become 4, 5, 6, and it will increase. So it will go down, it will go up. Okay. No change in ATP production is not going to happen. There will be increase in ATP production. So if you check the statements, okay, the first meant decrease glucose, decrease in sugar breakdown. This is true. Okay. Decrease in carbon dioxide evolution. This is false. Okay. Increase in carbon dioxide evolution. This is true. Decrease in ADP level per unit time. This is true. No change in ADP. This is false. Okay. So there are three statements. Statement number 1, statement number 3, and statement number 4 are true. Okay. So B contain 3 statements. Okay. So that means option will be B. Okay. Then see you in next video.